Okay. So my mom. Did we do nucleic acids yesterday? We didn't finish the presentation. No, just right. Nucleic acids is next week. We just did proteins. And we're going to finish proteins today and start learning from a new PowerPoint that talks about specifically about enzymes. And then next week we do nucleic acids and we're done. We're done everything. You're done the first unit of biology and the SPS next week. We just have this lab today, your project, the unit test next week, and that's it. Okay, so how we're doing, 20 people. We'll wait for a few more people to come. Just make sure you write down all the questions you have, all the things you're not sure about, primary structure, secondary structure. I'll quickly go over that. I'll also do the enzymes, and I'll tell you all about the activity we're doing for tomorrow. So while we're waiting for people, just to clarify, tomorrow's activity is 4 marks. It counts. It's the first time you will do something that counts for marks, okay? It's in Explore Learning, and that one, Khadija, you're fun. And that one you'll do in Explore Learning. Now, I'm going to check my trusty little note that I made, because some people in this class are not in Explore Learning yet. Who is it? Jusra, you're not yet, right? Are you in Explore Learning, Jusra? Have you done a login and everything? Let me check. That answers my question. Okay, so let me go. So we've been using this application called Explore Learning for a while, but you joined my class late, so I don't think you've done it. And on Monday when I gave that assignment, I don't think you actually did it. Yeah, the gizmo. Are you in? Oh, let me check if you're in my class. Yes, you are, but but you need to change it, because it has to be, your username cannot be your name. It has to be your student number. So I'm going to erase you and you log in again, okay? You register again. I'll give you, this is a class registration. I'll erase you and you do it again, okay? Who else is in that situation? There was somebody here that put a username that didn't, wait. Khadija, gotcha. You put a password that is not the right password, as did Tisha, who put twice the wrong password. Tisha, and also Ethan, and Risha. Look at this, how many people. Rachel also. Instructions, people. Aren't you in grade 12? Yeah, you're supposed to, your password must be your computer password. So I'm going to erase these following people so you fixed it, okay? I'm going to erase Tisha, I'm going to erase Ethan, I'm going to erase Richa, I'm going to erase Khadija, okay? If your username, the student number is already in use, you can put at the end of the student number an asterisk, a little star. You understand?
Wait, Miss, are you talking about Khadija with the K?
Yeah, honey, your username is your student number, but your password, is it your computer password? Because that doesn't look like a computer password.
Wait, I have to go check on good.
And Tisha, are you here, Tisha? you use password twice. Something to do with cake. You must love cake. But But Tisha, you have to use your computer password. You cannot use. I love cake myself, but you cannot use that. Okay? You use. You know the computer password that the school gave you that you use for everything in your school. You use that one.
Yeah, my password is my school password.
Oh, oh, wow, those are different then. Okay, so I'm wrong, Khadija. In my
It's
school,
like letters
they
and then
never
like
have that
two
one.
numbers, right?
Yeah, yeah. No, don't worry, sweetheart. In my school, they don't look like that, so I was surprised.
Yeah, I was so confused. In my school, they all look like that.
Okay, don't worry. Then that's fine. Tisha, I am pretty sure yours is not. It's not, so you can fix it. Okay, so I'm going to erase. I'm just going to erase Tisha and Ethan. Ethan, are you here? Fix that, okay?
Yeah, wait, I'm confused. How did I do it wrong? I asked you before.
Because your password is not your.
Okay,
Yeah.
that's the problem.
And then Risha, are you here? Because you also are wrong.
Miss, it wouldn't let me put a star beside my username because it said you can't use special characters, but can I put an underscore?
Yeah, absolutely.
Okay, thanks.
No worries. Okay. So I think we're all good now. So if, oh, and Rachel. Rachel, are you here? I don't know if Rachel's here. Awesome, Jusra. Let me see. Okay. So Jusra is sorted. Simabu is sorted. Hiva is okay. Okay, so I'm going to say your name if you're okay, and let me know if I didn't say your name because then we're in trouble. Okay, so Simavir is fine, Hiva is fine, Emily is fine, Sebastian is fine, Nicole is fine, Olivia is fine, Hazel, you're good, Andrea is good, Grace, Khadija, Oseme, Juliana, Rihanna, Sami, Derek, Tirija, Natasha, Zoe, Kesia, Kevin, Sunny, and Sean is all good. And then let me upgrade, what's it called, refresh, because then Jusra is going to be here as well. Brilliant. So if I didn't say your name, text in here so that I can check. If I didn't say your name, let me know. So let me see. I have now, right now, I have 23 enrollment, but our class has 25. So there is 2 people still missing. Who's missing? Let me see. Oh, Ethan is probably redoing it. Let me check Ethan. Yeah, now we're good. I don't know what, this thing is not in alpha. I don't know why. Ethan, you're good, and then Risha is good, and perfect. Yeah, we're perfect. Don't hack me, please. What does that mean? I don't even know. Okay, is Risha, you are here, yeah. Yeah. Yeah, good. Perfect. because tomorrow you don't want to waste time now and my login doesn't work, blah blah blah. So now you know your username, your password, you know how to log in, we're all good, we don't waste time, because the activity we're doing tomorrow takes about 50 minutes, 50, to complete. It's in Explore Learning, but it's not one of the little 5-question gizmos. It's a 50 minutes activity. So because it takes so long, you will use the whole period for this in case, in this class, actually, nobody has this problem, but in my other class, some people have Wi-Fi that is quite crappy. Sometimes Sebastian has this problem, where it gets kicked out and comes back. And so if that happens to you, and you're in the middle of the gizmo, you will lose time and progress, and then you have to come back. So if that occurs to you, I can see exactly when you get kicked out. I have a record. So you cannot pretend that you get kicked out. I know exactly when it happens. So if that occurs to you, you will get a bit of chance to make up for it, to come back and do it, okay? So, but otherwise you will be able to start and finish all within this period, and you'll be done, okay? So I'll explain right now what it is about, and you'll get to ask me questions as well after we do the lesson. Okay? So nobody has a terrible Wi-Fi right now, right? Nobody is in that situation, I don't think. Okay, so the activity you're doing tomorrow is a little bit like a case study. What I say is like a case study. So for instance, for my grade 11, sometimes I do things like this, or for you guys, I will do things like this, where you've learned everything about a topic, and now I will give you a problem that you have to solve using all your knowledge. So the case study tomorrow will be like somebody has some kind of disorder, some kind of problem where an enzyme doesn't work. And after the lesson today, when you learn everything about enzymes, so to finish with proteins, we're going to learn about how enzymes work. You're going to use all your knowledge to solve the problem. Now, the way you are evaluated is there's a rubric that Explore Learning provides, and I will use it to mark you. What are the things they look for? When you mark for communications, that your information that you put in there is concise, so it's not to worry, you don't write the whole essay, but you just get to the point and write scientifically to address a problem, okay? So you're succinct, in other words. You're not like all over the place, you're organized. So take your time, organize your ideas. If necessary, make a bit of a draft before you put it so you get good marks for communication. For inquiry or thinking and understanding, do I support my ideas with evidence? So if I've been doing this work and there's evidence that this problem exists, I use that evidence to support what I say, okay? So it's, in other words, it's an inquiry activity where I'm thinking scientifically, everything that we've been practicing. I put together all the information I've been learning about enzymes and proteins, and I use it to solve this problem. So it's consistent with the way I teach you. If you notice from day one, I've been teaching you this way. I say, why do you guys think this happened? Why does this occur? So I'm always teaching you with inquiry in mind. So now you're going to use all that knowledge to answer this. So they will give you. So the gizmo will provide you with a case study with evidence and experiments they do, and you'll be like, oh, this suggests this and this suggests that, and then you will produce hypotheses and answers and discussions based on that. Yeah, Olivia, it's all about enzymes and proteins. So, and on that note, I wanted to finish what we started yesterday, finish about proteins and finish teaching you about enzymes. I put already the recorded version of the lesson I'm going to do in your Google Classroom in case you wanted to review it. But before I start, does anybody have any questions about the primary structure, secondary structure, tertiary, quaternary structure, anything that you want me to address? Would you like me to briefly review aspects of that before we go on? No worries. I'll go over it briefly. No worries. Okay. Okay, let's do that. I'll go over, review it, go over the presentation, and we'll meet again afterwards to ask any questions you have, okay? So let's do that. So there's 2 PowerPoints that are involved, and I'll show you which ones they are so you can They are both in the Google Classroom. The first one is the one that we've been using all along, the one that is um, about, is this working? Yeah. So the first PowerPoint is the one that is macromolecules that we've been using all along. Okay, so I'll open it right now. And the other one is about enzymes. Okay, so let's start from the beginning briefly to go over proteins so that we know what we're doing. So in proteins, what we learned was that proteins do most jobs in the body. They have many important functions, and they are built up with three main structures, sometimes a fourth structure. The first structure of any protein is called the polypeptide, which is the long chain made up by amino acids that are joined by peptide bonds, okay? And that is what you see here in this drawing. Okay, so that's the primary structure of the protein, the polypeptide, the long chain of amino acids that are joined together by peptide bonds. Those amino acids that make the polypeptide, the long polypeptide will eventually fold into either or, and this is a typo, by the way, it should be alpha helix or beta pleated sheet. Those two can occur. So the same polypeptide may have a little bit of it that is in alpha helix and a little bit of it of beta pleated sheet. You can see it in the picture, and the picture shows you this, right? Some of it one way, some of it the other. Okay, so that's the second structure that makes up the protein. It's like a layer situation. I think it was Johnny that said this yesterday. It's like layers. You have first the primary, then the secondary, and then you have the tertiary. And the tertiary occurs because the R groups left over still haven't done anything in the primary and secondary. So what they do is they form connections that make the 3D shape of the molecule. They give the whole molecule its actual form. So these connections can be hydrophobic or hydrogen bonds. They can be covalent, like in a disulfide bridge, or even ionic when you have basic and acidic R groups. And all those make the 3D shape of the molecule. That makes most proteins. So most proteins will have primary, secondary, and tertiary. Some proteins will have quaternary. So some proteins will assemble the tertiary structures into a fourth form called the quaternary structure, and you have something like in hemoglobin, for instance, you'll have the 4 subunits, or glycogen contains 3 subunits. Yeah? So those are the structures of proteins, the 4 structures. So before I go on, does anybody have any questions about that part? All right. We're good? Okay, so to continue, that's what makes the whole protein. Now, this example that we discussed about sickle cell anemia is basically to illustrate that if the polypeptide has just the one amino acid different, that will change the whole shape of the tertiary structure, because the tertiary structure depends directly on the polypeptide. The R groups that will connect and make the tertiary structure will be related to whatever I have there. So if I have an R group with a different functional structure, then of course the shape will be changed. And in the end, this connects to this idea that the protein structure is completely consequence of the polypeptide. And if you alter that structure, we say that that's denature, that the protein has lost its nature. And what does this job in the cell? In the cell, when the cell synthesizes the whole polypeptide, which you'll learn how that happens in the third unit, then it goes inside this structure called a chaperonin, okay, which is, it looks like it'll be like a cylinder. This is the real picture, by the way. The first one is a real electron microscope picture. and then it puts pressure and it folds the polypeptide into its final shape. So this is how your body does it. But you will learn how the polypeptide is sequenced in the third unit. Okay? So that's the end of proteins in this PowerPoint. That brings the end of proteins for this PowerPoint. We'll do nucleic acids on Monday, and what we're going to learn now is these enzymes, how enzymes work. So before I go on to enzymes, who has questions? About the sickle cell disease, what is the importance of it? So we are just simply using it to show that if one amino acid is different in the whole hemoglobin, just one amino acid difference will make a completely different shape, and it will make a completely different structure of the whole protein, to the point that even the red blood cells look different. So it's just one amino acid difference in the sequence. It's to illustrate how the polypeptide determines everything else. Okay, anybody else has any more questions before we venture into enzymes? Because enzymes requires your knowledge of protein to understand the next part. That's in our protein. That is about protein. You mean the next PowerPoint? It's about enzymes, which is one of the jobs that proteins do. And Natasha said, well, we're about to talk about enzymes, which is a continuation of proteins, but it's in a different PowerPoint. Natasha said, can you all go over the last part? You mean what I just said about denaturation? Yeah, no problem. So in denaturation, so let me go back. So what I was showing you, so when proteins are synthesized, so your body will make a polypeptide, and the folding of the polypeptide will lead to the third structure, right? So you have the primary, secondary, and tertiary. So that is created in a molecule called a chaperonin, which is basically, it acts like this. It looks like a little container. And when the lid goes on, it creates such pressure that the folding of the polypeptide will look like the final protein folded. So these chaperonins exist in nature in our body, and this is how the polypeptide that is built in our body will acquire the final shape. When you throw in the polypeptide and put pressure, it always folds the same way, because the folding is directly dependent on the sequence of amino acids. Okay? So do we understand that part of the chaperonin? They are like containers. They are real things in our body, but they look like a container. Yeah, like little mini water bottles. I know, it's so cool. And it folds the chaperonin. The chaperonin, the polypeptide comes in, and when the chaperonin closes, the pressure that is putting on the polypeptide allows it to fold in the right shape. So now the next part is simply telling you how the proteins now do the job of being enzymes. That's the next PowerPoint. This only is about when the protein loses its shape for whatever reason. We call that denaturation, because it's lost its natural shape. Or if it were to return to its original shape, we say renaturation. Okay, so before I go to enzymes, but that's your body. Your body has all sorts of cool proteins that do cool jobs. I mean, you should go to YouTube after my class and look at an animation of what a motion protein looks like. and you would not believe it. It basically walks. It's just so cool. So denaturation is when the folding has changed and the protein is all loose, and renaturation is when it goes back to its natural shape. And good question, is the container a protein? I think it is. I don't know a lot about chaperonins, but yeah, I think it's its own version of an enzyme. Yeah. And it folds the protein. Yeah. Okay. So yeah, so cool. We need to know these for tomorrow. Not the chaperonins. The chaperonins we will need only in the third unit. But the presentation I'm about to do, you will need for tomorrow. Now, Olivia is asking, so is the helix and beta pleated sheet the natural shape? Well, okay, so when we say denaturation, what we mean is the tertiary structure, those are the secondary structure. The tertiary structure is no longer working and you've got the loose polypeptide. So that means you've lost all of the other shapes too, the secondary as well. You just have the string loose. Maybe a little bit of the secondary is left, but the tertiary is done, which gives the shape to the protein, okay? Okay, let's go and look at the enzymes presentation, which is what you will need also for tomorrow. So you need both the knowledge of enzymes, the knowledge of proteins, and the knowledge of enzymes. So enzymes, as you know, proteins do many jobs. And by the way, can you see this? So proteins do many jobs. The job of proteins as enzymes, we're going to pay specific attention because we need that for the next unit. So the next unit is about how we change molecules to extract their energy or to make molecules. So we need the knowledge from this unit to build out the second unit. So enzymes are very important in doing these jobs because enzymes are natural catalysts. So we need to pause and learn about enzymes quite a bit because this is a job we care about a lot, more than maybe other jobs we will learn later. So enzymes have a few things they do. First of all, as you know, they speed up reactions. Okay, they are like natural catalysts, and reactions can be divided into two major groups of reactions, anabolic and catabolic. So you knew, for instance, we say decomposition or synthesis. So a dehydration is a synthesis, hydrolysis is a decomposition, right? But when we have a number of reactions that eventually lead us to make something new, instead of saying a synthesis, we say those reactions are anabolic. So now you just saw that. Did you see that? You guys see that? That's my husband forgetting that I'm teaching, asking for lunch. I'm glad I'm not recording this. Okay, so as I was saying, catabolic reactions is another way of saying decomposition, but we say catabolic because instead of one reaction, you have a number of reactions that break down stuff. We say anabolic when you have a number of reactions that make something new. Okay, so that's the term we use. Instead of synthesis or decomposition, we say anabolic, catabolic. Cool? Now, enzymes are almost always made of protein. That's why we are bothering learning about them. There is some exceptions, like sometimes you have something called ribozymes, which are enzymes made of RNA. But don't worry about that yet. And the other thing that matters is that enzymes may have like a sidekick or an assistant that helps them function, and those are called coenzymes or cofactors. So they kind of join the enzyme to help it work. You will see more of that next unit. And vitamins are that. So when they say, oh, you must take vitamins, it's because, not because they are nourishment for you, but because they help you function. They help your enzymes do their jobs. Okay? I just thought of a very cool question I want to ask you. I don't want to forget. Okay. Now, when we say function of enzymes, first of all, important thing, enzymes are in small amounts in our body. They're not that abundant. They're in small amounts, and they are very specific. Okay, so every reaction has its specific enzyme that will connect to that specific reactant. It's not like a universal thing. And they don't get consumed in the process. They just help in the reaction, but they are not reactants or products. They are just simply helping. And they are not going to change the thermodynamics of the reaction. You probably are thinking, what is she meaning with that? I'll show you in a minute, because you probably don't understand this. And they require specific pH concentrations and temperature to be at a certain level, and I'll explain. And these things can denature, these very conditions can denature the protein, and I'll show you what I mean. So before we go on to this, which is the thermodynamics, does anybody have any questions?
Face now.
Yeah, I'm waiting, Olivia. No, they are like helping the reaction, but they are not reactants and not products, so they are not used up. Yeah, we'll discuss that in a minute. Yeah, we'll discuss that in a minute. Yeah. So this is an analysis of the reaction as a change of energy. Okay, so you can look at a reaction and think of it as in thermodynamics, from the point of view of thermodynamics. And what that means is, how does the energy of the reaction change? So what you see here is the reactants have a lot more energy than the products. As time passes and the reactants convert into products, the reactants have a lot more energy than the products. So my question to you is, why would the reactants have more energy than the products, guys? What would be happening there? How come? Okay, some energy is lost, but that sounds like a huge amount of energy you've lost. Okay, so you're saying, Seme said, it takes more energy to create something new. Since the products here have less energy, then you didn't make something new then, by that logic, Oseme. You didn't make something new? Why the products have less energy? Something to do with the bonds, says Tirija. So if the reactants have this much energy and the products have less energy, what did they do with the bonds? So the reactants had, exactly, so the reactants had more bonds than the products. And that's why the products have less energy, right? So what kind of reaction was that? Anabolic or catabolic? Why is it catabolic? Because the products are what? What did I do? Exactly, nice job. You guys are on the ball, nicely done, exactly. So I took something that was big, like say that I had glycogen, and I cut it, and then my product is a bunch of glucose. So I took this big molecule, cut the bonds, my product is a bunch of glucose. So all those bonds release energy, therefore my products now have a lot less energy. Does that make sense? Okay, so we understand the reaction and we understand catabolic. Khadija, you almost always understand, so don't be surprised. So the reactants break bonds. Yeah, exactly. The reactants used to be a big thing, and now your reaction broke them apart, and you got a final product. So, do you think this reaction is a dehydration or a hydrolysis? If I had to choose between those two, exactly, hydrolysis. You cut off all the bonds, and you had a bunch of little things as a product. Nice. So, in this reaction that I have here, what occurs is we want to understand how is it that the enzyme achieves this goal. So the reactants are these things, the products are these things. Normally, the reaction will require all this energy in the white curve here, all that energy called activation energy to make the change occur. But if you have an enzyme, the reaction requires a little bit less energy to get the change to occur. You have still the same reactants and the same products, and the enzyme doesn't get consumed. It just makes the change easier. Okay, think of it as this. Hopefully you guys clean your room better than in that example that we learned the other day, where the kid was flinging clothes to, to clean the room. So if you clean in your room and you go with a broom and you're cleaning, that will take you quite a bit of time. Eventually the room will look clean and it started being messy. If instead of doing that you use a vacuum cleaner, you will do the same thing. You will achieve the same result, but it's less effort. You have more help. The enzyme does that. The enzyme is like the vacuum cleaner. Yeah, that makes sense. Nice. Okay, so that is what's happening. The enzyme is helping the reaction, but we want to understand how is it that the enzyme does this. So this is a perfect example. So here you have a polysaccharide, like you know, okay, this is a starch, and this is what the enzyme does. The enzyme has a perfect fitness in shape with the reactant. They fit perfectly in there. So it's easier for the reaction to work and cut off the bond because they are now connected to a base, and that base makes it easier for it to occur. Okay, you want to think of it as, I don't know if you've ever done some work, construction or things like that. I would have to, for instance, cut wood or even a loaf of bread. You have to cut a loaf of bread. It wouldn't be as easy to cut it if you held it like this than if you have it on a table and hold it carefully and then you cut. So that makes the change much easier because it's connected to that base that is the enzyme. Okay, so this is how the enzyme helps. So things that we want to know about the enzymes. Why is it that the reactions don't occur without the enzymes? Well, they could occur, but they would be so slow. So the enzymes have all sorts of advantages that allow the reaction to occur fast. First of all, the enzymes can connect to the substrate, which is another name for reactant, perfectly because they have the same kind of shape. So they fit like a key in a keyhole. They connect perfectly, okay? And then the other thing that occurs is that because the enzymes are so big, it's easier, for instance, for two parts to meet each other or for the bonds to become unstable and break. Okay, so between the fitness and the shape becomes much easier. The same when you have two things that have to come together. In this example, for instance, you see the two reactants fit perfectly, and then it's easier for them to join because they are now in a stabilizing environment, let's put it that way. It stabilizes the transition. Okay, and that is how the enzyme work. And there's a few terms to understand, which is the active site, the part that connects to the reactant, substrate, which is another name for the reactant, okay, and everything else is pretty much the same. So before I say a few more things, any questions about the enzyme and how it works? Okay, perfect. Oh, can you explain the catalyzed part again? Yeah, so what occurs is that this substrate, which is another name for reactant, will connect to the enzyme in the section of the enzyme that fits perfectly the shape of the substrate, which we call the active site. And when it connects, because the enzyme is big and it maintains also the stability of that substrate, the changes can happen easier. If the bond has to break, the bonds will be easier to break because this helps in separating them. And if they have to come together, the two molecules that have to come together, because they fit so perfectly, now they can connect more easily. So the fact that the enzyme is so big and it fits the shape of the substrate helps, okay? And this is another way of seeing an analogy. This is a colleague of mine that gave me this picture. think of something that manages to break things apart with a lot of strength, okay? It just helps, makes the bonds break more easily, okay? Now, what occurs with the enzymes is enzymes have what's known as optimum conditions or ideal conditions, okay? So there is a rate of the reaction and there's an ideal temperature at which the enzyme will work. Now, here's my question for you guys. Yeah, that's probably steroids. I'm guessing. Yes, Khadija, a specific enzymes for every specific reaction. Yeah, absolutely specific. Every enzyme has to fit a specific substrate. And that's why nothing else can help. Like if you're lactose intolerant and you don't have lactase, the enzyme lactase that breaks down the lactose, which is a sugar in milk, there's nothing you can do without that enzyme. Another enzyme cannot take over. You need that very enzyme. Otherwise, right? So that's what happens. Okay, so I was asking you, so temperature has an ideal temperature. Every enzyme has an ideal temperature at which they work best. My question is, why do you think the temperature has an ideal point? And then if you go lower or higher, the enzyme won't work anymore. Why do you think that is? What do you think is happening?
Can you repeat the question?
Sure. So we said that there's an ideal temperature at which the enzyme works best. And if you are too high or too low the temperature, the enzyme will no longer work. Why do you think the temperature, how is the temperature not helping the enzyme work? What is it doing? How is it affecting the enzyme that no longer works? It must affect the shape. Good guess, Olivia. Maybe it's denaturing it. Why? How is it changing the shape or expanding it, like Simaver said, or changing? Why is it denaturing? Why is the enzyme, what's happening inside? Remember, this is a protein. So it must be affecting the bonds. Which bonds give the shape to the protein? Which specific structure is being affected? Do you think it's a peptide bond? What's the first structure that is likely to suffer from the temperature effects? Is it going to be the peptide bond? What will be the first thing that you start losing to the nature? Amino acid is the same as saying peptide bond. If you say hydrogen, you're talking about the secondary structure. I don't know what you mean with water. Of all the structures that make up the protein, when I apply the temperature and I now have a more loose chain, what am I losing? Am I losing the peptide bond, or what am I losing when the shape is changing? Exactly, Olivia. I am affecting the R group interactions. Before you worry about I destroy the chain, that eventually may occur. Or I may destroy eventually the alpha helix and beta pleated sheet. Before I get to that point, the first thing that will happen is the R group's interaction will be affected and the shape will be changed. Right? It's like, remember, it's like layers. Okay? Like Shoni said yesterday, it's like layers. You first have your food, then you put your sock, then you put your shoe. Polypeptide, then alpha and beta, then tertiary. So the first thing that is affected is tertiary, then exactly. Yeah. It's the same, okay? You put on your sock, then your shoe, you take off your shoe, then your sock. Okay, let's keep on going. So that's what occurs with the temperature. You have to have an ideal temperature. Now, before I tell you more, what else do you think could affect these R interactions? What other things could happen to this protein that now all of a sudden the shape is messed up? What else could mess up in my R interaction? Think of how the R interactions happen. So you know your R interactions is the first things that will unassemble. So temperature can mess it up. What else would you add? And it would now all of a sudden the R interactions are being affected. What else could it be? That you add and now the R interactions are not working and it's denaturing. pH, said a bunch of people. pH, what do you mean with pH? Explain yourself. I know I said pH earlier, but what does that mean? So, anionic concentration. Okay, let's do pH first. What does it mean, pH? Why pH? You're right, Olivia, but let's think about what that means. Why is it that you know that in the right pH you are altering the shape? What is happening? Think of everything you know about the R interactions, about altering the pH. What have I added? Have I changed the amino acid molecules? Wait, Terja. We said it's the R interactions. That's not the amino acids yet. We're talking about the tertiary structure. So how am I affecting? Okay, so disulfide bridges? Could be, sure, but this is a more obvious one. More obvious R group. What R group would be affected if you add an acid or a base? Yeah, so if I had the acid or basic R groups and I threw in there an acid or a base, or not the nonpolar, because the nonpolar are not going to react with an acid or a base, right? They're going to be inert to that. But the polar stuff, so cool, hydroxyl and sulfur would, but before that, the acid and basic R groups will definitely react exactly. The amino and carboxyl group R groups, not the of the 100 amino acid, but the R groups will be connecting to the acid or base that you added and changed the pH, right? And that will alter the R interactions and it will alter the shape. Okay, third option, which over here, Natasha wrote. She said, and ionic concentration. Why is that going to denature my protein? Ionic concentration. Why? Well, the name tells you. So what do you mean? Explain. You can do it. You can do it. You added something that is ionic. What is that? What is an ionic thing? Guys, you know what? Think of cooking. More than polarity. It's ionic. Polar is covalent. It's ionic. What is an ionic thing that you can add? Think of cooking. Yeah, you add salt. What would happen if you add salt? What is the salt doing?
Face, man. I want you to make baby face.
And what is it going to do with the R groups?
How?
And what will happen to all the R groups?
Oh. Look my line.
Well, they won't become polar. They simply will be attracted to the ions that you added and leave the connection they had to the other R groups, right? So if I had something like OH, and OH was making a hydrogen bond here, well, it's like when you add salt to water. Immediately the hydrogen bonds start connecting with the ions and all the ions dissipate. So this is going to separate and start connecting with the ions, and the whole shape of the protein will change. And they will be a different shape. So think of this. Think of how we cook. Okay? When we cook meat, what do? We use what? Of these three things, we can use salt to cook meat. Yeah, you can cook meat by salting it, right? Like that's what people do when they smoke things, or sometimes they just cook simply raw stuff by adding salt. What else do you use that we mentioned here? Yeah, but what else? Not just oil. Oil and what? Heat. So temperature, salt. Do we ever use acid to cook? Do you cook things in vinegar? Lemon juice. Yeah, so we're doing all those three things to proteins. That's what we're doing. We're denaturing the proteins in food. So when you put salt or tenderizer for meat to make it soft, what you're doing is destroying the proteins to make it soft. Or when you're cooking the steaks or cooking food, you are using temperature to destroy those proteins and tenderize them and make them soft. And when you use vinegar or lemon, or you make ceviche or things like that, that's also what you're doing. Okay, so when you use lemon juice or vinegar, you are also doing that to protein. To repeat the explanation of pH. So when you are adding an acid or a base, what you are doing is having the acid or the base connect with the acid or bases that are in the R groups of some of the amino acids. And that is going to interfere with the R group interaction that connected the acids and bases to make the 3D shape. And it's going to change the shape of the protein. Exactly. So when you add lemon, and sometimes people cook raw food with these things. Okay, sushi is raw, but you add a lot of soy. So that's helping sort of cook. Good question. So you clean raw chicken with vinegar, does that denature the protein? The problem with raw chicken, I'll go over this quickly before we go back because I don't want to miss. The problem with raw chicken is raw chicken has a microorganism called salmonella. And salmonella, even if you were destroying some of the stuff, produces toxins that make you very sick. So even if you were somehow getting the chicken smoked or cooked, you have to be very careful because salmonella can make you quite sick, even if you were taking care of it. So that's the danger. Okay, cool. So I'm going to go back to the PowerPoint to continue. I agree, Zoe, and to clarify a few more things. So basically, this has at the end a few questions that help you. One is, for instance, what's the active site, the enzyme, the section of the enzyme, or the substrate? Or in the induced fit model, the shape of the enzyme when the substrate binds stays the same. And the answers for this is a section of the enzyme, and the enzyme will adapt to the shape of the substrate, so it will fit, okay? Now, another set of factors that affect the enzyme function is not just temperature and pH, like we said earlier, but there's also things that can interfere with the function of the enzyme. So for instance, another example of things that interfere with the function of the enzyme is if I have enough substrate or enough enzyme. So I always use this analogy when there wasn't COVID and you would go to the movies. When you go to the movies, there's always a lineup in the washroom for females and no lineup in the washroom for males. And the reason is think of it as how much substrate there is for enzyme. For females, there's a lot more time that you spend using the washroom. So there's always a lineup. For males, there's less time using the washroom, so there's no lineup. And the people that design these things don't realize they need to make more washrooms for females, so this problem doesn't exist. So the same occurs with enzymes and reactants. If you have too much of the substrate, the enzymes, there'll be a lineup to use the enzyme. If you have too much of the enzyme, there won't be enough substrate to use it up. It's a ratio thing, okay? That's one factor. The other factor that could affect, as I said, was pH. And finally, the things that can interfere with the enzymes, these are practice questions, are inhibitors. So these are molecules that can slow down the function of the enzyme. So there's one kind called competitive inhibitor. They call competitive because they compete with a substrate. So they may have the same shape of the and then the reactant, and so they fit the molecule, the enzyme, very well, and they compete with the substrate, so the molecule becomes slower because it cannot change the substrate because there's something else taking the space. Your body sometimes naturally uses this ability. Like for instance, if you ate too much food, your ATP, remember? ATP, the energy you made, will make the enzymes work down slower. They will act as a competitor against glucose, and the enzymes will work slower. And there's also non-competitive inhibitors called allosteric inhibitors, because allosteric means shape. What they do is they connect to another section of the enzyme, and the enzyme now has changed the shape, and it may not longer work. Some poisons are like this. They ruin the enzymes in your body, and you start getting very sick because you cannot work anymore. But there's also something called allosteric activators, which are small molecules that join the enzymes and sort of like switch them on. They activate them, so the enzyme won't be the right shape until the activator joins. So they work kind of like this, but the opposite way. And your body has a few examples of allosteric activators that sort of switch on the enzyme. Okay? So basically what we're saying is some things can be competitive inhibitors because they look like a substrate, and some things can be non-competitive or allosteric inhibitors because they change the shape of the enzyme. Okay? Who has more questions? So Khadija, substrate is this name we give to the reactant in enzymology, okay? So it's basically what the enzyme will alter or change into the product. So non-competitive inhibitors or allosteric inhibitors are inhibitors that will change the shape of the enzyme forever. Now, when you sometimes have non-competitive activators, those use the same principle, but what they do is they change the enzyme in order for it to work. Okay, so that's a side note. See, that's why it says allosteric activators also exist. So they work in the same principle, but what they are doing is instead of damaging the enzyme, they kind of switch it to the right shape. Exactly. It's, yeah, good analogy. Yeah, like they're remodeling. So if we have some enzymes in our body that, for instance, only when the activator is on, the enzyme is kind of working, and if the activator is not on, it's inactive. So it allows us to control the function of the enzyme. Yeah, exactly. It's like a switch. Cool. Yeah, it kind of sits there until it's switched on, basically. Okay, so that's the end of enzymes, and that leads me to, now we can discuss any questions you have for tomorrow. So again, you will arrive to class, log into the Explore Learning, and you will be given 50 minutes to work on it. There'll be questions that require you to answer, explain your question, explain your answers, take your time, write carefully, don't overwrite, don't write too much, get to the point, use evidence so that you get a good mark. How pH affects enzymes again, lol, I couldn't write everything. Don't worry, don't forget, Olivia, everything I say is recorded and you can go back after class and write it properly, okay? So the goal is not to write now. The goal is to listen, and then you can always organize your notes after. But pH affects enzymes because when you add an acid or a base, that is not the ideal pH for that enzyme. That means that they are connecting to the R groups that are acidic or basic in the tertiary structure and interfering with those bonds. So now the acid and the base that were connected, you added something in there and it started messing up that bond, and now those connections will break and the enzyme will change its shape. Sure, Shani. So anabolic means when you have a number of reactions that eventually synthesize something. And catabolic means that you have a number of reactions that eventually will break down something. So anabolic is similar to saying synthesis, only the synthesis is one reaction. And anabolic, you may have many reactions. Catabolic, same than saying decomposition, but you have many reactions to break down something instead of just the one reaction. Okay, more questions about everything we've done? I would love to hear any questions about what we're doing tomorrow, anything you want me to go over. Just to confirm again, the gizmo tomorrow is based on everything on macromolecules, PowerPoint. No, just proteins and enzymes. Just proteins and enzymes, only that. It's really about enzymes. I mean, of course, you have to know basic macromolecules, but it's mostly about enzymes. It's really about enzymes. So make sure you go over what we learned today. You remember catabolic, anabolic, energy, all this stuff. Well, something like that. You will be given a problem, and basically you have to apply your thinking, inquiry skills that you've been developing with me. Okay, so every time I teach you, if you notice, I'm asking you questions, I'm asking you to connect ideas, to put them together. This is what you're doing now tomorrow. You're going to be asked questions and you have to connect all the evidence, take your time and write down why do you think this suggests this is the problem. Okay, it's almost like you're a detective of the body. Okay, if you want, I'll give you an example of grade 11. What if you're not finished in 50 minutes? Well, it is designed to be done between 40 to 50 minutes. So it's designed that way, it's made for high school. So it's expected to be done that way. Now, if you have a lot of Wi-Fi, and I can see it, I can see if you have Wi-Fi issues, then you have a bit of extra time because our classes are 75 minutes. Or if you're a student that is entitled because you have either your ESL or you have an IEP and you're entitled to extra time, you will be given extra time. But if neither of those three things occur, you are not somebody with an IEP, you are not ESL, you're not entitled to extra time, you don't have Wi-Fi issues, you have to finish in the right time. Is there going to be a Google Meet? Yeah, we'll meet here and I will be here. I won't leave you because if you have any problems, I'll be here to answer your questions, okay? Not to answer the questions of the actual assignment, but to answer questions if you have trouble. This is, yeah, I know, Shanice, I can't wait. Yeah, don't worry. If you need a bit extra time, if you're ESL and you're entitled, I'll know. But I was going to tell you an example, for instance, in grade 11, my students, for instance, they would be given a case study in person, like when we were in person. So they were learning the digestive system, and I would say, so a person shows up in a doctor's clinic and says, I have chronic diarrhea. So you would have to analyze everything you know about your digestive system, what is happening, put it together and answer that question. My gizmo is full of bad quiz marks, haha. Well, okay, so many students in this class have that problem, partially because they're rushing when they do the gizmo. They quickly do the gizmo and then do the quiz without taking their time. This gizmo is not like that. You don't have this thing. So take your time when you answer this stuff. Take your time when you're writing your answers. If anything, you can even write a draft and then do it, okay? The lineups of the washer. So I was saying that the proportion of reactant or substrate and enzyme has to be right. Otherwise, it happens like when we go to the washroom and there's females and males. If you have too much substrate and not enough enzyme, this will line up, right? Because the proportion has to be such that the substrate can be used, product, and then there's not the delay, the line up. So that's another factor that can affect the enzymatic function. Exactly, in the pacing, right? Exactly. Okay, so we still have a bit of extra time, so please, if you have any questions for tomorrow, anything that you don't know, do ask. We'll meet here, and then we'll go to explore learning. Who else has any questions about everything? The 4 structures of proteins, enzymes, anything. Okay, before we leave, so the quaternary structure is what exists in only some of the proteins, and when they have it means that you have more than one tertiary structure that joins together. Not exactly. They may ask you, it's not like that. They may ask you, what's your hypothesis? What's the problem? It's a combination between inquiry, lab skills, and solving problems. Okay, I'm going to quickly double check that who is here. I think the one tomorrow doesn't have any section of multiple choice. I forget. They sometimes have a section of multiple choice, but they are not like the gizmos at all. They're different. I'm just going to double check that everybody's here for a minute to make sure. So I know Jusra is here, Simabir is here. Is Hiva, you're here, right? Hiva's here. Emily, are you here? Yeah, okay. Nicole is here. We just talked. Olivia. I just want to make sure that everybody knows what they're doing for tomorrow because I worry. Andrea, are you here? Okay, perfect. Just checking. Grace.
I'm here.
Okay, I know Khadija is here because we talked a minute ago. Oseme is here because we talked a minute ago. Juliana, I don't remember. Are you here? Okay, so Juliana maybe not. Rihanna, are you here? Rihanna is here, okay. And then Sammy, are you here? Sammy is here. Sonny said he's here even though I didn't ask. Derek. No worries, Sonny. Derek, are you here? So Derek, I don't know. Okay, because I have to double check because I don't want anybody to get caught tomorrow and then they don't know. Tisha, I know we were talking earlier. Ethan, we were talking earlier. Richa, we were talking earlier. Thirisha, are you here, Thirisha? Yeah, you're here. Who else? Natasha, we talked a minute ago. Zoe. Kesia, are you here? Perfect. And then Kevin, are you here?
I'm gonna have to tell my teacher tomorrow that I have an ESL, or if it's called, so I get extra time.
So Kevin, no. Okay, so Juliana, Derek, or Kevin are not here. Oh, Derek's here. My bad. And Kevin is here. What about Juliana? Okay. And everybody knows how to get to Explore Learning Now Pro, right? Okay, I can't wait to mark this, okay, people? I cannot wait. I can't wait. I hope all of you will be amazing. No, don't be scared. Just review your notes, write it all down, review all the stuff we've done, write it all down, make sure you understand it, okay? Have confidence that you can think on your feet because you've done it all through the lessons all this time, okay? So just take your time, and that's all you need to do, okay? Okay, guys, well, lovely to see you. Can't wait till tomorrow. Just review your notes, study, and you should be fine. Cool? See you tomorrow.